Basic Science lecture for this academic year. It is a pleasure and an honor to introduce today Dr. Andreas Papassotiropoulos, who is a board-certified psychiatrist and psychotherapist. And since 2007, he's a full professor of molecular neurosciences at the University of Basel in Switzerland. His area of expertise includes investigation of the molecular basis of human cognition and the development of improved therapies for neuropsychiatric disorders. And his research combines targeted and genome-wide genetic, epigenetic, and transcriptomic analysis with functional brain imaging in healthy and diseased human population. He's the recipient of numerous honors and awards, amongst others the Robert Bing Prize, the Weizmann Lecture, and the Cloetta Prize. And I think we're all very happy to welcome him today. So with no further ado, I will pass the microphone to Dr. Papassotiropoulos.
So thank you very much, Dimitri. Thank you very much, everybody, for the honoring invitation. I'm really very happy to present in the framework of this initiative, of your initiative, which I really think it is. So today, now let me share my screen. Today I will be talking about some of the work of our lab, which is targeted towards developing better drugs and better therapeutic options for neuropsychiatric disorders. And it is a genome-based approach that we follow. So a few words about our division. So since I came to Basel, we managed to build up a department that it is truly transdisciplinary. So we combine colleagues from various disciplines, all of them working together for one goal, and the goal is to develop better therapies. So we are based on methods such as human genetics, which we combine with deep phenotyping in large samples, in large populations that are deeply phenotyped for cognitive and emotional traits, with behavioral tests, EEG, structural, functional MRI, TMS, transcranial magnetic stimulation, etc. But we also do need molecular biology to explain some of the human genetic findings, because as you very nicely know, human genetics is correlative in its very nature. So we do need biology to explain some of these findings. Since a couple of years, we also include in our portfolio virtual and augmented reality, and everything with the aim to develop and to do clinical trials in our division, but also in collaboration with others. So we are based in the Department of Psychiatry, in the Biozentrum in Basel, and in the Department of Psychology. It's really truly interdisciplinary. So the work I will show today is a common work of many colleagues and postdocs and doctoral students in my division. From all of them, I absolutely would like to refer to Dominic Dequervin, with whom I share the entire endeavor. Dominic is the chair of the Division of Cognitive Neurosciences. I'm the chair of the Division of Molecular Neurosciences. So we really combined our groups with the goal I stated before. So let me begin with what is the reason, what is the rationale for performing human genetics in neuropsychiatry? So the rationale is very clear. Neuropsychiatric disorders are heritable, full stop. So we're talking about large amounts of heritability, like, for example, schizophrenia, 80%, Alzheimer's disease, 70%, major depression, 40%, anxiety disorders, also 40%, autism, 90%. So neuropsychiatric disorders are heritable, and because this is the case, it is legitimate to use human genetics for a variety of reasons. For example, to discover susceptibility genes, and by this, to try to describe molecular pathways of the disorders of interest. And ultimately, the wish is to use human genetics to identify biology-driven and human biology-driven drug targets. In the last couple of years, there has been a tremendous progress in human genetics, which is due mostly to the conduct of very large genome-wide association studies in the field, which can be done due to the organization of very large consortia for specific disorders. So there has been a tremendous progress in human genetics, but there has been less progress on the phenotypic level. And to me, genetics stands and falls with the choice of the phenotype. And here is something where psychiatry, but also neurology, has a problem, in particular psychiatry, and this is something which was a motivation to me to start the work I will show to you today. So two examples for the problematic phenotypes in psychiatry. I assume you perfectly know neuropsychiatric diagnoses are, in fact, we're talking about spectrum of disorders, and they are characterized by substantial phenotypic and biological heterogeneity. And the phenotypic heterogeneity can be a tremendous problem. For example, you can diagnose a patient with major depression. If that person suffers from significant weight loss, insomnia, and psychomotor agitation. At the same time, you can diagnose another person with major depression when that person suffers from significant weight gain, hypersomnia, and psychomotor retardation. Two completely different clinical pictures, one diagnosis. And this can be a tremendous problem for any research, for genetic research, for enrollment in clinical trials, etc., etc. How about another disease, which is, let's say, if you wish, more clear phenotypically? How about Alzheimer's disease? Again, the same problem. If you just take one method to characterize a correlate of Alzheimer's disease, which is structural brain imaging, you readily realize that also there we do have a tremendous amount of phenotypic heterogeneity. This is an MRI of the typical AD with cortical atrophy, enlargement of the ventricles, and of course, engagement of the hippocampi of both sides. This is another MRI with a so-called limbic predominant Alzheimer's disease, where mostly the limbic system is involved. Here you have the completely opposite, the hippocampal sparing AD, where the hippocampus, at least structurally, is not yet involved. And here we have another example of Alzheimer's disease with minimal atrophy. So all of these subjects have been subjects in genetic trials, and they do have 4 different phenotypical pictures of the same gross disease. So the phenotypic heterogeneity in neuropsychiatry is a huge problem. And in fact, what can be a solution to this problem? The solution is to use precise phenotypes, but how exactly to do this? So first of all, one has to realize most neuropsychiatric disorders are not dichotomous categories. What we are talking about is about a normal distribution of traits in the population, with mental disease representing the extremes of the distribution on multiple dimensions. It can be cognitive dimensions, emotional dimensions, etc. So we're talking about the extremes of a phenotypic distribution, which is a normal distribution in the population. And Dominic and I have really very early proposed in neuropsychiatry to step back a little bit, not to do genetics or research only on disorders like, for example, schizophrenia, but try to deconstruct neuropsychiatric disorders into biologically meaningful domains. And we proposed that such domains should fulfill at least following criteria. First of all, they should represent physiological domains, so domains that you can measure in the general population, cognitive domains, emotional domains, which are known to be disturbed in neuropsychiatric diseases. The second criterion is such domains must have specific and testable neural correlates. For example, as shown in human brain imaging studies. Why? Because we don't want to stay only on the behavioral, observational, or self-report level. And the third requirement is such biologically meaningful domains should be heritable, because otherwise we cannot use genetics for that. So we're talking about domain-centered research. So which domains to use? One of the domains is episodic memory. Why is episodic memory a biologically meaningful domain? First of all, it is a physiological capacity that can be measured in the general population. It is impaired classically in Alzheimer's disease. It has a neural correlate, for example, atrophy of the hippocampus in the medial temporal lobe, and episodic memory is heritable. How to test episodic memory in humans? So it is more or less trivial. What you have to do is, for example, to apply episodic memory tests, such as word lists that probants have to learn, and after minutes or hours to do, for example, a free recall of the learned words. This here gap is very important. Depending on the delay you apply, you really study different molecular pathways of the episodic memory trait. For example, after many hours, this is really truly protein dependent, whereas after minutes, it's not really dependent, the capacity upon the production of new proteins. Another way to test episodic memory, and why I'm showing this because I will refer to this test later on. Another way to test episodic memory is again it's not to use words, but to use pictures that probants have to encode, to learn, and then to freely recall minutes or hours later. And we show such standardized pictures. For example, this very nice one picture, this more neutral picture, this emotionally negative picture, or this emotionally positive picture. And why I'm underlying the issue of emotionality here, because episodic memory performance is not independent on the emotions that are elicited during learning. And there are different types of episodic memory, depending on whether the learning material is emotionally neutral, emotionally positive, or emotionally negative. And with such tests, we can really very nicely deconstruct and fine-grain the specific episodic memory traits we're interested in. What we end up with is, and this is a true histogram of one of our studies, where we studied the aversive memory capacity, so the capacity of individuals to preferentially remember negative emotional material. And this is a true histogram. This is raw data, if you wish. And you see here, this is a very nice normal distribution in a large population of healthy young adults. So you end up with exactly what you really think or expect, what you measure, which is most of the probants are okay in their performance. Some of them are extremely good, some of them are extremely bad, but this is normal physiological variability in the population. And this is a variability we need in order to combine this heritability, this phenotypic variability, with genetic variability. And in this case, of episodic memory, we do know that the heritability of episodic memory performance is around 50%. So the genome really occurs a large amount of variance of this variability. And very trivially, what then one has to do is to look for polymorphisms, to stratify the population here genetically into different genotype groups, and then to test for statistical significant differences between groups. So why was for us human genetics so important? We started around 2000 with our research and genetic research on human cognition. And at that time, already a lot was known about cellular and molecular correlates of memory. For example, here Kandel's very nice review, 2001 in Science, which described a lot of molecules that are known to play a role in synaptic plasticity, a really very established correlate of memory capacity. You know all of these molecules here. The issue is this knowledge was already there, but it was taken from animal studies, from snails, rats, mice, etc. And we asked ourselves, how can we be sure that this knowledge can be translated to humans? In other words, how can we be sure that a molecule that is known to be important for the gene withdrawal reflex of a snail, a very basic learning mechanism, how can we be sure that this very molecule is also important for humans? One established method to do so is human genetics. And we started, I'm showing really this graph because it's a historic graph. Dominic came to me, it was 2000, he had published a paper on the influence of cortisol on human memory. And this was a test he applied to the population. Each dot is a proband. Here probants had to learn 60 words and to recall them freely 24 hours later. So what you see here is most of the probants remembered some 15 to 18 words after 24 hours. These guys here, and these are really male individuals, remembered really only a few words. And this lady here remembered even 52 words out of 60, which is tremendous. So, but then here we realize this is physiological variability, what we are looking at. And we knew at the same time, the heritability is large. So how about looking for genes that explain partially this variability? And we started, remember, it was 2000, with a classical candidate gene approach. And we focused on the serotonin 2A receptor gene because it was amply known that the serotonin 2A receptor is important for memory in animals. And most importantly, in humans, this gene is polymorphic and it harbors a functionally relevant amino acid substitution, with the tyrosine allele being less functional. So we recruited a population of individuals and we performed 3 different tasks. One, non-hippocampus dependent, which is immediate recall of words. This task is not dependent on the function of hippocampus. And then the 5 and 24 hours later, delayed recall, which is dependent on hippocampus. And what we realized was, yes, the tyrosine allele is associated with worse episodic memory performance after 5 minutes and 24 hours. And this was the first study, together with another study of Danny Weinberg at NIMH, that also was published that year in 2003 in Cell on the BDNF gene. So these 2 findings were the first to, let's say, establish human genetics of physiological human cognition and episodic memory. Then we went further, and what we here did a couple of years later, we did not focus on only one candidate gene, but we basically wrote down a list of candidate genes, all of them being important for synaptic plasticity and appearing in papers that were reporting this in animal studies. And what you see here is among all of these genes, the genes highlighted red were found to be significantly associated with episodic memory performance in our population. But not only this, a combination of these genes, and if you wish, a linear combination of alleles and an individual genetic score which reveals how many of the alleles each individual is carrying that are associated with better episodic memory, that individual polygenic score was associated not only with episodic memory per se, but with the function of hippocampus during an episodic memory task. The more the polygenic risk score was, the more activation we observed in the medial temporal lobe in the subjects during the episodic memory task. This year, we already had embarked on another approach, because remember, this was still candidate gene dependent approach, but we had 2005, 2006, it was the early era of genome-wide association studies, and we were the first to perform a GWAS on episodic memory performance in healthy young subjects. And by this, we were able to identify polymorphisms within the KIBRA gene. KIBRA stands for kidney and brain. I didn't know of the existence of this gene until then. KIBRA polymorphisms were found to be highly significantly associated with episodic memory performance in this population. Because we didn't know anything about KIBRA, we then went a little bit further and we realized that KIBRA is expressed both in humans and in animals, mostly in the hippocampus in the temporal lobe, a region where you would expect a gene to be expressed if it is related to episodic memory. That KIBRA genetic variants were also associated with differential brain activity, specifically in the hippocampus and parahippocampal gyrus during an episodic memory task. So these findings established a correlation and association between KIBRA and episodic memory in humans. But then this is, of course, now a very important step. In the candidate gene era, we were dependent on pre-existing knowledge from biology to take our candidate genes, to nominate them. Here, we just produced unknown associations and, let's say, correlations. And now, this is a knowledge we can give further to biologists to establish, hopefully, a functional rationale for this association. And this happened in the group at Johns Hopkins, in Rick Huganir's group, who, driven by our findings, produced KIBRA knockout mice. And what he realized in different memory paradigms in animals, that KIBRA knockout mice showed diminished or reduced memory, or let's say phenotypes that are memory correlates in these animals, based on 3 different behavioral tasks. So this was the first time that we really saw a translation of the human genetic findings back to animals. GWAS can be, of course, are nice, but still they just lead or result in simple gene lists, as if genes work in isolation, which they don't. So further developments of the GWAS approaches were related to the fact that one now could take not only single SNPs and test for association with a certain trait, but to take gene pathways or gene sets. And what the test does, it looks for a combination of association signals in a certain predefined pathway. And there are many predefined pathways out there according to the pathway list, or let's say pathway website you use. But here, it is a different concept. Here we're talking about the significance of a whole pathway, not just of single genes. So here we used this approach, the gene set or gene pathway approach, to ask whether the findings we get from genetics of episodic memory in healthy individuals can translate to disease. So what we here did is we tested episodic memory in two larger samples in Basel with a pictorial task I've shown you before. And by doing this and the subsequent replication studies, we ended up to only one out of all examined known predefined genetic pathways that was really significantly associated with episodic memory as a pathway in healthy young individuals. And this pathway is the calcium signaling pathway, which of course harbors a lot of genes. Then we realized that this pathway was not only related to episodic memory as a behavioral task, but was also related to activation of the hippocampus in a large sample of fMRI tested individuals, over 1,000 in our lab. So the calcium signaling pathway and the genes therein are associated as a whole to both a behavioral task, episodic memory, and activity of the brain during an episodic memory task. And this activity is specifically located in the hippocampus, where you expect this to be. But then, does this knowledge translate to disease? And the short answer is absolutely. Among all of the genes of the calcium signaling pathway, we discovered a core set of these genes that was associated with not only episodic memory in healthy young subjects. This set of core genes was associated with episodic memory in another replication, small sample of healthy young subjects in Zurich. It was also associated with episodic memory in a sample of aged individuals, non-demented, in Germany. And it was also associated with the risk for Alzheimer's disease in a large GWAS study. So yes, what we found here by starting on episodic memory performance in health, it can be translated to disease afterwards. So this core gene set is associated with episodic memory from physiology to pathophysiology. Still, these are nice findings and correlations, but we lack functionality. We really don't know why. And of course, we cannot do this for each and every gene we describe in the course of the GWAS, but for some genes which seem to be very interesting, we do use in our lab an animal model of learning and memory, which is based on C. elegans. And here, this was one of the, let's say, the first finding we used, which is related to translation of our human genetic findings to animals. During the course of our human genetic studies, we realized that there is one gene, Musashi, that showed a very nice association signal with episodic memory performance, with recall of with pre-recall of memory. But we didn't know why. So what we did is we tested this in C. elegans. And how to do this in C. elegans? So C. elegans is a very nice animal. The nice thing about C. elegans is it can learn and it can forget. So how to do this? It's a very classical conditioning example. So the worms are eating bacteria and on this plate. This is what they do all the time. Then you can, if you add a chemoattractant, they are attracted, which is normal. However, if you combine this chemoattractant with something the worms hate, which is starvation, they then learn that this chemoattractant is in fact something bad. So if you put then the worms that learned this association of chemoattractant with starvation, if you put the worms back on the plate where they can start eating again, and you put the otherwise chemoattractant, they go away from the chemoattractant. So this is a learning process, and you can really nicely quantify it. And we have shown that, for example, so these are N2 strains, so wild type worms, if you wish. So they're attracted by the chemoattractant, but then they learn to avoid it after 1 hour if you combine this with starvation. And here are worms where we knocked out the glutamate receptor. This was a positive control. Glutamate receptor should be involved in memory in worms, and absolutely it does. So here, glutamate receptor knockout worms, they are attracted normally, but they forget very quickly to this association between the chemoattractant and starvation. So it was a positive example, the positive control. How about Musashi knockout worms? Musashi knockout worms, so first of all, wild-type worms. First of all, wild-type worms here, they, after 10 minutes, they go away, so they are not attracted by diacetyl. But then over the course of 60 minutes, they forget it. So they show this curve and they still begin to be attracted again. Not so Musashi knockouts. Musashi knockout worms learn as efficiently as wild-type worms, but they forget less. So Musashi knockout worms have a forgetting deficit, which is important. So to cut a long story short, why this happens, so Musashi is a translational inhibitor, and among others, it inhibits the translation and production of up to 3 complex, and it leads to, by this, to reduce actin branching, and so to loss of synaptic size increase upon learning. But if you block Musashi 1, or if you knock out it, then you don't have this inhibition of translation, and then you still have large synapses, and we have shown this. This model here allows for us C. elegans-based pharmacology. So here we took the known Musashi pharmacological Musashi inhibitor gossypol, which is otherwise a toxic substance. We cannot give this in humans, but what we saw here is, and this is important, these are humanized C. elegans strains. So this C. elegans strain here is a knockout Musashi 1 with knock-in of the human Musashi sequence. So they are humanized worms. So these worms, if you just give them DMSO, they forget after 24 hours. So they begin to go back to the chemoattractant. But if they are pre-treated with gossypol in different concentrations, they forget it. So they have a forgetting deficit, which might be important in the course of for diseases that are known to perhaps to profit from a forgetting deficit, for example, Alzheimer's disease. So here we have established that we can inhibit pharmacologically Musashi, and it improves memory in C. elegans. And now we embarked on a high throughput screen with 90,000 compounds to look for Musashi inhibitors that we can test, and perhaps even after a couple of years, hopefully, also give to humans. Back to the domain-centered research, I've talked a lot about episodic memory, but here we have another cognitive domain that it is equally important to us. It is working memory. So working memory is a classical memory capacity that is a physiological memory capacity of human beings. We know working memory is impaired in schizophrenia typically, but also in Alzheimer's disease. Working memory has typical neural correlates, for example, activity in the dorsolateral prefrontal cortex, and working memory is significantly heritable. So a perfect domain, cognitive domain that fulfills all of the criteria we described before, so that we can take it as a core trait to do genetics with the hope that these genetics are translatable to disease and perhaps to lead to results to new drugs. So first of all, if we study the genetics of working memory in healthy humans, Does this knowledge translate to disease, to schizophrenia? Exactly the same question that we asked before with episodic memory. The answer is, again, absolutely yes. So here what we did is we did a genome-wide association study of working memory capacity in healthy young adults. We used the gene set or gene pathway approaches I presented to you before. And among all of the gene sets, there was one gene set that was really replicably in many samples associated with working memory capacity in healthy young subjects. And the pathway is the voltage-gated cation channel activity pathway. So genes of this pathway were significantly associated with working memory capacity, and the entire group was associated, the entire path was associated with working memory capacity. We also established that a polygenic score of these genes, of the cation channel activity pathway genes, is also highly significantly associated not only with behavior, so with working memory capacity, but also with the activity of the brain during a working memory task, mostly in the parietal lobe and in the cerebellum, two known regions for working memory capacity. And most importantly, this polygenic score of the voltage-gated cation channel genes was also associated with the risk for schizophrenia in a large schizophrenia consortium sample. So yes, this knowledge can be translated to disease, which is important because it leads us to what we are now doing, is to use genetics to provide human genetic evidence for novel drug targets. And what is our concept? First of all, we use the mega GWAS of psychiatric disorders and mega GWAS of relevant traits, like, for example, cognition or educational attainment in already in publicly available GWAS. The problem with these GWAS here is the respective phenotypes are blurry. This is, for example, here a disorder phenotype. Schizophrenia, yes, no, is blurry by itself. But with our database and with our approach, we have the ability to filter here those gene loci that are also important for intermediate phenotypes, for cognitive domains, as for example, working memory, episodic memory, or others that are in our hands. And by this, hopefully, to nominate genes and gene products that are relevant and that are perhaps druggable. So the basic concept when you do so is following. You start with a GWAS of a certain trait. And this GWAS here, the results, they provide you essentially with a list of genes that are associated with a disease. For most of the genes and the gene products, no compounds are known. So you would have to do a drug design and to do safety proof of concept studies. So you have to go all the way through a classical drug development approach. You have to be Novartis or Roche or Pfizer to achieve this goal. In your list here of interest, out of your GWAS, there might be some compounds that are known already, but they are not used in humans. So again, more or less, you don't have to do drug design, but still you have to do tox studies, etc. Again, you have to be big pharma. But how about if you are lucky? How about if some of the genes here you identify and their gene products, perhaps they point to drugs that are already on the market, but are used for other indication? Then you can readily take these drugs and repurpose it. So, and people have shown that yes, the genome is druggable, human genome, and it can be used for repurposing approaches. For example, using genetics, one realizes that drugs that are used for cardiovascular diseases, they can be also used perhaps for mental disorders or for other diseases, etc., etc. So yes, the human genome is druggable or points to druggable pathways and to repurposing pathways. to be honest, we've shown this before in a clinical trial. In fact, what we had done, and this was back in 2013, we had started with a classical GWAS of a physiological memory capacity, which was aversive episodic memory. So the preferential ability to remember bad emotional material. And by doing a genome-wide study in this large population of healthy young adults, we had nominated two pathways that seem to be related to aversive memory, the long-term depression gene pathway and the neuroactive ligand receptor interaction pathway. Essentially, again, a list of genes. And one of the genes here was a histamine receptor, which we found it is important because we thought we can readily use antihistaminergic drugs because the genetic findings nominated the histamine receptor as being important. So we did use diphenhydramine, an old antihistaminergic drug that passes the blood-brain barrier, and we performed a phase 2 clinical trial with a primary endpoint aversive memory. In other words, does a compound that was nominated through a GWAS on aversive memory, does this compound really affect the very phenotype, aversive memory, in a clinical trial subsequently? The answer was again, absolutely. Use of diphenhydramine led to significant reduction of negative, of aversive memory, but did not lead to any reduction of positive episodic memory or of neutral episodic memory. So diphenhydramine did just not dampen the cognitive capacity of the subjects. It led to a specific decrease of aversive memory capacity as predicted by the GWAS we performed before. So now we took this concept and this here is something exciting. We just received very generous funding by the Swiss National Science Foundation, what we call the reset approach. So what we do here is we know there are around 1,400 genes that are targeted by, or gene products that are targeted by already approved and known drugs or compounds. So 1,400 gene products. Then what we do is we do a GWAS of a disease that is of interest, and with a question, how many out of these genes are related to that particular risk for that particular disease? Then, if we find certain number of genes, then we do the important filtering. So how many of these genes are also associated with important intermediate phenotypes for that particular disorder? And then we hope to find one or two that we can test in clinical trials. So here we are now. We did this for schizophrenia. Here we identified 104 genes out of these genes here that are associated with the risk for schizophrenia. And out of these 104 genes, 2 of these, under strict Bonferroni correction, 2 of these are also associated with working memory performance. So these 2 genes correspond to known drugs because they started from here. So we can perhaps look at the drugs and see whether we can test them in clinical trials. What are these drugs? It's streptozotocin. It's a chemotherapeutic, which is not available in Switzerland. And it's dalfampridine for aminoparidine, a very known drug, which is available in Switzerland. So we thought this might be important. So remember, this drug was nominated out of this entire GWAS concept and filtering steps we predefined. So a couple of words about fampridine or for aminoparidine. So the respective gene that nominated fampridine is a common hit in schizophrenia and working memory genome-wide association studies. We're talking about potassium channel blockers. potassium channels, and famotidine is a potassium channel blocker. It improves walking speed in some adults with multiple sclerosis. The mode of action of famotidine seems to be more or less known. It improves axonal conductance, but it also improves rapid neurotransmitter release. Some studies have shown that famotidine might improve cognition in multiple sclerosis, although the results are not really conclusive. It's highly permeable through the blood-brain barrier, and it's formulated as a prolonged disease tablet, and you can give 10 milligrams twice daily. So a perfect drug for us to repurpose. So now we already started a study in healthy young subjects with famotidine. We also now additionally already scheduled studies in patients with schizophrenia and also major depression. We are talking about double-blind, placebo-controlled crossover design studies, repeated dose, with primary outcome working memory performance. In other words, does this drug, which was nominated out of the working memory in schizophrenia GWAS, does this drug improve working memory performance in healthy young adults or in patients with schizophrenia or major depression? Of course, we have some secondary outcomes, and we include 44 subjects. Remember, it's a crossover trial, so the same subjects are also taking placebo. So it is as if you had a parallel design study with 80 individuals, but a crossover design trial is, of course, much better because we have the internal control. Each subject is its own control. So I'm absolutely fascinated by this. I do believe that genome-guided drug discovery will lead to some important findings and some drugs that can either be repurposed, or, of course, it will lead also to new compounds. But we are an academic institution. These compounds can be only, if they are new, can be only developed through collaboration with pharma. A word of caution, though. First of all, this procedure here will not work without real commitment. So if you only half-heartedly do this, you will fail. And some pharmaceutical companies have started or had started to use such approaches, but really it was half-heartedly. So this is a problem here. You really need commitment. Expectation management is important. Not all targets based on human genetics will succeed. And it has a variety of reasons why they will not succeed. But expectation management is truly important. Exchange of experience is also equally important through different groups. So because there are a lot of ways of how to do this, and a lot of filtering steps you can apply. And last but not least, it is about collaboration on a common goal that will hopefully change the therapeutic field in neuropsychiatry. And talking about collaboration, and this is my very last slide, this is something we very pleasantly do. I mean, the aim of our department is really to study the molecular underpinnings of cognitive and emotional traits that are related to health and disease, with the overall goal to develop drugs against neuropsychiatric disorders. So for this, we have already a very nice resource. We have really a unique database of deeply phenotype subjects, a unique database of psychometric information, plus corresponding genetic, epigenetic, transcriptomic, metabolomic, and brain imaging data. So we're talking about a population of 4,000 subjects, all of them, of course, with genome-wide arrays, many of them with functional and structural brain scans. Importantly, all of these people, 1,800, were scanned in one scanner. So it's extremely homogeneous sample. And this scanner in Basel did not undergo any software or hardware update, which is very important for the signal. Out of these, we have already 1,400 whole exomes available. metabolomes, methylomes, a little bit of transcriptomes, and importantly, we have serum and plasma from these subjects stored that we can use. So everybody who wants to collaborate, we can collaborate, among others, on the basis of this very rich resource that has already shown to produce clinically relevant findings, and I do hope that it will continue to do so, either only through our group or through collaboration. So thank you very much for your attention. I hope I've presented something that might be of interest also to basic researchers, but to clinicians too. I didn't show all of the results and activities of our division. For this time wouldn't be sufficient, but in any case, thank you very much for your attention. I'm looking forward to take all of your questions.
Andrea, thank you very much. That was fascinating, I must say. Let me start with a question. So you're doing those pathway-specific polygenic risk scores. I wonder if you think that this type of approach could be used to distinguish patients that might be indistinguishable clinically, but might have different pathways involved in their disease?
So yes, with a question mark. So first of all, I think it is very important to think about if such an approach is feasible, where exactly, so in which disease would it be feasible? Psychiatrists, of course, try already to use polygenic risk scores to, let's say, to characterize patients better. The point is, whereas the significance, we're talking about highly significant results, they come from large-scale GWASs where the effect size is very, very low. So if anything, such polygenic risk scores will be important at the extremes. So you have to take the extreme high and extremely low polygenic scored patients, and then subsequently see, is there any difference? And where exactly is the difference on the phenotypic level? Which means you really need also deeply phenotyped patients, not only schizophrenia, you know. But let me come to something. So I think yes, but it is a lot of work to do, and still psychiatric genetics has to prove that it will have any implication in the clinic beyond what I've shown, let's say, nominating drugs. So until now, psychiatric genetics didn't have any implication for the clinic, but the field is pretty new. Talking about Alzheimer's disease, I'm extremely still really enthusiastic about that finding of calcium signaling genes because, and this is now something important, Alzheimer's disease is not a disease, is not a memory disorder per se. And this is very important to realize. Alzheimer's disease is a neurodegenerative disorder that happens to occur in a memory important brain region. But Alzheimer's disease is a neurodegenerative disorder. It's not a disorder of disturbed memory processes. However, some of the memory processes can lead to cognitive reserve, so can, let's say, phenotypically mask neurodegeneration. And I think exactly this is where, for example, calcium signaling genes might play a role. Of course, calcium signaling is a ubiquitous molecular event, and it plays a role in many things. But what I'm talking about is, I do believe that a subset of Alzheimer's disease has to do with reduced ability of resilience, of phenotypic resilience to the disorder. And so some people still have good firing neurons, or I mean, they degenerate, but they still fire very nicely. So they mask phenotypically Alzheimer's disease, and some not. So I believe that a subphenotyping of Alzheimer's disease patients according to their polygenic scores on several pathways will be important. And I do believe that calcium signaling will be one of these pathways.
Thank you. Any more questions for Dr. Papassotiropoulos?
I could, if I could ask, hi, really great presentation. I think this is very important for young students that they're following our seminars in HIAAD. So just a first question. Do you think that in that approaches with mega GWAS analysis, you're going to pick more of what we call coming from the stress or chronic stress background of Max Planck Institute of Psychiatry and all the work developed in both depression and the connection between depression and Alzheimer's disease? Do you think that you could also identify mechanisms of vulnerability or, let's say, resilience? I don't know, because some people believe that there are different mechanisms involved in one or the other, by the way. Especially in circuits that are, let's say, the bridge circuits between depression and AD. And I'm talking not geriatric depression, like really early, as a first signs, because more and more we see in Alzheimer's disease patients that more and more of them, like 60 or 70%, they even have some kind of neuropsychiatric problems before the appearance of the real AD, let's say, profile.
So thank you for this really important question. And the short answer is, again, yes. I have to say, nevertheless, that although we use, and I use results of mega GWAS, I'm cautious when using the results of mega GWAS just because of the granularity of the phenotype and the fact that the phenotypes used in the mega GWAS are really very blurry. Having said that, so that you really have to be cautious about that, I absolutely believe that there is at least one pathway that is important both for stress and for the development of stress-related psychiatric manifestations, plus it really plays a role in neurodegeneration. And this one pathway is the glucocorticoid pathway. And we have shown by ourselves that genetics of glucocorticoid pathway really show, let's say, lead to the identification of genes of the glucocorticoid pathway that are related to stress-related disorders and to phenotypes also in healthy individuals that are that show a normal distribution, phenotypes that are stress-related. So, so the glucocorticoid pathway is one of the classical pathways I would directly jump in and look for genetic alterations. Plus, there might be others. I think the next step would be more an agnostic step, because when we're talking about pathway-related research, we use predefined pathways, KEGG pathways, gene ontology pathways, and whatever pathways. And they are predefined based on some criteria. On the other hand, we don't know, you can group the genes the other way around. So what we need is a more data-driven, let's say, agnostic grouping of these genes. But for this, you need machine learning approaches. And for this, you need big data. And with big data, I mean really big data, which then has to be phenotypically nicely done. Well, I don't think I will experience this in my lifetime, which I hope will be enough. But it is a problem. But to answer your question, yes, stress is a central denominator and a common denominator. And resilience to stress is a common denominator for many manifestations, including neurodegeneration. Absolutely.
Would you place this resilience on the GR signaling and genetic response to glucocorticoid receptor signaling in specific brain areas, or you think it's a more full organism level, because you were speaking about specific signals you got in the MRI analysis in specific brain areas that was part of the initiation or the establishment of a phenotype in a healthy population or later in a disease extrapolated. So what is your view on that? And what is your data?
So the view, it is a very tough one, what you're asking here. And I mean, we could talk about this for ages, honestly. If I try to be very short, so first of all, stress is, of course, a ubiquitous phenomenon. Stress is not brain related. It's, everything suffers under stress. But of course, there are some regions that particularly are affected by stress, particularly. And with this, I'm not talking about HPA axis. HPA axis is, let's say, the generator of everything. But for example, hippocampus, parahippocampal gyrus, this region is particularly affected by stress and by hypercortisolism. So, although I wouldn't abandon and forget other brain regions, I would really, this brain region and MTL, so medial temporal lobe, hippocampus is one region I would absolutely go for. Our data shows, the genetic data shows that genes that are related to, so the glucocorticoid related genes are associated with aversive memory capacity and are associated with PTSD, with post-traumatic stress disorder in different populations, and are associated with the function of the hippocampus. And also others have shown this too. I mean, we're not the only ones. If you take Elizabeth Binder's work at the MPE in Munich and others, I mean, there's a lot of literature. So I think focusing on this pathway is important. And by using such rich data sets as ours or others, can contribute a little bit to the answer of your question, but just partially.
Thank you.
So if I could ask one more question, you mentioned PTSD, so I can't help myself but to ask about the gene Musashi, I guess it was called, in C. elegans, that was that controlled aversive memory. Is there a human homologue, and is there any connection to PTSD?
So absolutely. So not to PTSD yet, but the point, and this is important, Musashi, the human homologue is Musashi 2. We have, in humans, we have MSI1 and MSI2. The real close human homologue of the C. elegans Musashi is Musashi 2. Musashi 2 in our hands was really nicely, significantly associated with long-term free recall. So after 24 hours. The point is, in humans, you don't know why you see this association. Do you see this because the probants learned better, stored better, or retrieved better? You just see the final output. So you don't know why exactly this happens. In worms, we realized Musashi is an activator, is an active participant of forgetting. So forgetting or synaptic shrinkage upon learning is an active process. It's not a passive thing. It's not just decay. So, and in fact, Musashi is for us now, hence now, a very important finding because it's one of the genes that we described that is really specifically related with forgetting pathway. So then you can say, well, if this is the case, it might, if you forget more, on the one hand, it is bad because you forget and you have an Alzheimer's-like phenotype. But if you forget, this might be good for disorders where you should really forget, which is PTSD. And it is desirable to forget. So we didn't see any association of Musashi with risk for PTSD. We see, but because this is not specific to emotions. So Musashi leads to an overall forgetting, irrespectively of the emotional content. And I think this might be one reason why it's not related to PTSD, among other reasons. But we have described other genes that are related to memory and, let's say, of written only emotional material, and that are also associated with risk for PTSD. One of these genes is the alpha to beta adrenergic receptor. This is one, I didn't show this finding now, it's one of the most replicated findings in psychiatric genetics. This gene is related to amygdala function, is related to learning and forgetting of emotional material specifically, and it is also associated with symptoms of PTSD in PTSD patients. So yeah, but Musashi not. Still, Musashi is a tremendously important finding, and this is also why we now perform a high throughput screen, compound screen, to identify Musashi inhibitors. But then I can tell you, if we identify Musashi inhibitors, we will have to test them biologically. And C. elegans will be one method to do so, but we rely, for other methods, we will need collaboration.
Thank you. Any more questions for Dr. Papassotiropoulos? Well, I will ask you one last one if nobody else is.
I have one for the RNA binding proteins, but if Dimitrios, if it's not, please go on.
No, it's just a quick question, really, about finding the druggable genes. And so you go through this two-step process of the GWAS here for schizophrenia and then working memory. You end up with, and I assume you end up with two variants, not two genes. So how do you go from those variants to the genes that you?
In fact, we end up with genes, and in fact, what we do are so-called gene-based tests. So we are not looking at one specific variant of a gene. We look at the entire gene, its entire genetic variability, and whether it is related to a phenotype of interest. Then the mechanism why that particular genetic variability is related to the phenotype, I mean, it is something, it is an R1 grant per gene, but I don't care. The point is, this gene nominates a drug.
But how about the direction of perturbation of the gene? Like up or down?
No idea. So then we rely on pre-existing literature. And if there is a pre-existing literature, then perhaps we can have an idea about direction of effect, whether antagonizing it or promoting its function or gain of function will be good for the particular phenotype, or you can just test it in humans. Give the drug and see what happens.
Vaso, can we take one more question from Yannis?
Okay, I hope it's short because I think we are running. We are already,
Just very fast because there are a lot of students from the masters, from the Athens Neuroscience master that Spiros Avramopoulos is running and listening this talk. So I think they will be important because these weeks also they are doing the the plasticity course, and it will be very important to listen your view whether you think that there are specific genes that can regulate synaptic plasticity related, for example, to emotional memory or emotions or depressions, or other genes for memory and synaptic loss involved in memory of associated memory, because that sounds a bit peculiar to me at least. So what is your opinion? Because what they learn in the classical books test is that synaptic plasticity doesn't have to do with the output, the behavior output. It happens there. It depends on the circuit and the area it happens. That's why we have a different behavior output. So what is your view based on this great work you're performing?
Well, I mean, the whole issue of synaptic plasticity is, of course, highly complicated. But if you ask me, I don't know, to put it this way, it is a testable hypothesis which you can do to see whether there is emotional input dependent synaptic plasticity.
Okay, now I got it.
Yeah.
Thank you.
You're welcome.
Thank you very much. That was really a great talk and very stimulating.
Thank you very much.
Yes, I would like also to thank Andreas for this very, very exciting talk. It was an honor to have you here. And I want to say, I thank you on behalf of HIAAD, of course. And I want to say the goal of HIAAD is to bring together scientists that they work on neurodegenerative diseases. I think we accomplished to do this. And whoever wants to talk to you, Andreas, more, they can definitely send you an email. They can definitely establish very nice collaborations. I saw from your last slide that you have beautiful resources and very important ones. And yeah, that's what I would like to say and encourage everyone to keep going with discussions and collaborations.
So thank you really very much for having me. It was an honor. As I just said, I'm absolutely open to collaboration. And allow me only one comment. I started my career as a young scientist, and I was really interested in impact factor. Really, it was. I mean, science was extremely important, but it was important the corresponding impact factor. I was not free from that. Now I'm not interested in that. I'm interested in impact without factor. And impact can be done through collaboration and through translation to clinics. So really, I'm open to collaboration. You can email me, whatever, WhatsApp me. I would be very happy to see whether we can do something together.
Thank you so much. That was the best way to close this great presentation. And I would like to thank everyone for attending the HIAAD Basic Seminar Series. Thank you very much.
Thank you very much, Andrea.
Γεια σας, Σπύρο.
Thank you very much.
Γεια σας, Σπύρο. Γεια σας, παιδιά. Γεια σας, ευχαριστώ.
Bye. Thank you.
Να είστε καλά.